Welcome and good afternoon, everyone. My name is Ludovica, and I'm the Outreach Coordinator of the Future Vaccine Manufacturing Research Hub, or Vax-Hub. I'm very happy to be here today to introduce you to the third webinar of the Platform Funding webinar series. Before starting, just a kind reminder that this webinar is recorded and public and will later be available on the Vax-Hub website and UCL Biochemical Engineering YouTube channel. So I would kindly ask all participants to keep your microphone and camera off during the presentation, but please do join the conversation on the chat using the chat. This said, I would like to start with a brief introduction, and then we'll move on with the presentation of the project. So, as I'm sure many of you already know, the Vax-Hub has been supporting innovative research related to the development, manufacturing, scaling up and manufacturing of vaccines, primarily of need for low and middle income countries. And so we decided to organize the Platform Funding webinar series in order to present to the public the great results that were obtained by several hub-funded feasibility studies. These were funded back in 2020 using part of the budget obtained by the Department of Health and Social Care. So today's webinar is on the validation of the MiniTEM system as an analytical quality control tool for vaccine and viral vector manufacturing. And it will present results from a collaborative hub-funded feasibility study that was awarded to Vironova and UCL Biochemical Engineering and conducted with the support of the UK's National Physical Laboratory. In this webinar, this webinar will introduce you to a validated negative transmission electron microscopy analytical method using artificial intelligence with acceptance criteria for analysis of virus-like particles, or VLPs. We will dedicate around 30 to 40 minutes to the presentation of this project, and then allow a 20-minute Q&A session, for which I would like to invite you all to send your questions using the chat. So it is now my great pleasure to introduce our speaker, Dr. Saba Hussein Gore from Vironova, where she is the head of strategic market and customer development. So Saba obtained a PhD in the structure and function of membrane proteins from the University of Cambridge, after which she pursued her research as a postdoctoral fellow at NYU and then at Stockholm University. So a very warm welcome to you, Saba. Thank you for being with us today. I would also like to introduce Professor Dan Bracewell, who is a professor in bioprocess analytics at UCL Biochemical Engineering and who will chair today's Q&A session. So welcome both. Thank you for being here, and welcome everyone who joined us today. And now over to you, Saba. Thank you.
Thank you so much. I hope you guys can hear me. I will share my screen and actually start the presentation. And let me see. And hi, Dan. I'm looking forward to having a nice Q&A session afterwards. So let's see. Let me know. Okay, like this and like this and like this. Right. And I will open my presentation here. Okay. Can everyone see my slide?
Yes.
Yes. Yep.
Great. Okay, thank you. So hopefully this presentation should take about 30 minutes. I'm really happy to sort of be presenting this on behalf of our collaboration. And as Vita mentioned, today I'll be talking to you guys about the MiniTEM system and how we've used it as an analytical quality control tool for VLPs. Let me now go to the next slide. That's good. So let me just give you a bit of an outline to what I'll be going through today. First, I'll start off with a super brief introduction to Vironova. Then I'll talk to you guys about transmission electron microscopy and how that is used in process development. I'll outline the project aims, the methodology, and the results that we obtained, and then sort of summarize it up, linking it to some of the relevance that we believe we can contribute to the vaccine manufacturing field. So Vironova, who are we? Well, we are a company that wants to bring sort of unique strengths to our customers by making the subvisible accessible. And we do this by providing electron microscopy and making that technique more accessible, either through our service, where we've got sort of vigorous GMP certificates available that you can kind of work around, or as an in-house transmission electron microscope that you can use for your sort of test in the lab. And both of these sort of systems and both of these setups comes with a pretty powerful software that I'll be talking to you guys about more. And our main application area for our TEM, or transmission electron microscope analysis, are within the fields of gene therapies, vaccines, and drug delivery particles. So some of you might be very familiar with TEM and how that's used in process development, but for those of you who aren't, I'd just like to give a bit of a summary of what we can do with TEM. So we use TEM at Vironova because it allows us to visualize things in the nanometer range. And that's great to get information about sample status and understand what's happening during the process development. And when I talk about this nanometer range, you know, here are the particles that I think a lot of us are very interested in or might be working with. So you can see things from AAVs, phages, lentiviral vectors, influenza, etc. And not only can you make these direct observations, but you can obtain really valuable information about not only the sample itself, but the actual particles. In addition, I want to point out that you need a low amount of your precious sample, so 3 microliters of your sample. The process to prepare the grids that are imaged in the transmission electron microscopes, and this is me talking about nsTEM, so this is room temperature transmission electron microscopy, requires very few consumables. The process is quick and it takes approximately 5 minutes. You basically add a sample onto a hydrophilized grid. You blot that off with some filter paper. You wash it with some water, and then you apply a stain. And I'll talk to you guys later about the stain. When you've done this, you leave your grid to dry, and you can actually store it at room temperature. And you can go back and image and re-image this grid at least for a year, if not more. And I quite like that, and I think users of TEM like to be able to go back and revisit grids and revisit their sample and pick up some new information if there's something that they want to kind of go back and see. But what do you get then from these 3 microliters? You can actually answer quite a lot of questions about your particle size distribution, purity, integrity, and morphology. And I want to show you a few examples of that. So for particle size distribution, what do I mean? Well, you can potentially pick up or look at what debris you have in your sample. That's your sample here to the left, where you can compare the amounts of debris in one sample versus another. You can also potentially pick up some specific hidden contaminants. Here we can see some proteasome contaminants in an AAV sample. You can also use TEM to look at your purity assessment to give you some informed, to allow you to take sort of informed decisions around your process development. And that can vary. It can be things like checking that your baculovirus is gone after your purification, etc. You can look at your particle integrity and what happens when you change, when you heat it up or you make some changes in your formulation buffer, and how that affects the sample morphology. You can also use it, of course, if you are using it routinely, to really keep an eye on your batch-to-batch variations and just kind of checking on your sample. It's very useful for quality control. Or you might be interested in looking at or picking out some specific morphological feature that might be linked with biological activity. Here we are looking at and tracking influenza VLPs. So I'm going to move this so that you can see. So these are influenza VLPs, and we are using transmission electron microscopy to keep an eye on the spikes and what happens with those. So I hope that that's given you, given you kind of a good set of examples to convince you that nsTEM insights do support better decisions in the process development. But despite this, it's not a technique that is frequently used and definitely not routinely used. And if you look at why, you can kind of understand, because there are quite a lot of hurdles to overcome to get to valuable TEM insights, or the data that I showed you, the graphs and the analysis you have on your sample. And let's break that down and kind of look at it step by step. So the first thing you need to do to have this method in-house is, of course, to bring in your electron microscope. And for these electron microscope to be installed, you need to often make some facility fittings changes. And that's quite pricey, and it requires a lot of work. Then when it comes to the training of the electron microscope has to be done on site. So you have to make sure that you've got the application specialist, and they need to be present for the training to take place. The sample preparation as well requires radioactive substance as a stain traditionally, and I'll get back to this later. It's great to have a method ready to start working on and some SOPs at hand, but there isn't really that much around, and especially not things that are really sort of heavily method validated. For the imaging and the analysis, you're quite dependent on an expert user. It's a complicated system and a complicated analysis. And in addition to that, this expert I've seen often is spent sitting and doing a lot of manual annotations to analyze the images that you take with the electron microscope. So that means that the dissemination takes time, and also your expert user is spending a lot of time on doing quite a mundane process. And of course, any kind of support needs to be done on site, which of course bring in a risk. So the project aims then, and what we saw was that when we joined Vax-Hub and we started going to these meetings, which is a great place to sort of exchange ideas, we saw that we had some complementary areas of expertise that would help us to potentially address these challenges in adopting TEM. And we wanted to focus on VLP analysis in this context and a focus on LMICs. So we did this together as part of the Vax-Hub feasibility study and the Vax-Hub interaction voucher as well, together with the National Physics Laboratory, MPL, and UCL. And why VLPs? If this is the Vax-Hub audience, then I don't think I need to go too much into detail. I actually took the information for this slide from a really nice newsletter that Vax-Hub had sent out, where the focus was on VLPs. And I've seen a lot of great presentations about how VLPs are really useful in development of vaccines. So I'll just briefly tell you, their size is very favorable. You can express them in a range of systems, including bacteria, insects, mammalian cell line, yeast, etc. Their surface geometry is organized in a sort of a repetitive structures, and it enables them to really get a good, strong cellular and humoral immune response. And of course, you can sort of modify and exploit the exterior and the interior surface to your kind of liking. So there's a lot of good things going for it. So what did we then bring to the table for this work? At Vironova, we brought our MiniTEM system. This is a small transmission electron microscope. Some of the key aspects of the MiniTEM system is that it's really compact. So it's very easy to install and to move. You don't need to change anything in your facilities or build anything or reinforce the floor. And you only plug it into sort of a normal wall socket as you would do with your phone. You can also use the system. So as soon as you've put in your sample, all of the imaging and analysis can be done remotely, which is extremely valuable. And I'll get to that in a minute. It's also packed with a lot of automated functions. So we can get away from this dependence on an expert user. And the software itself here helps with speeding up the image acquisition time as well as the image analysis. And here we want to show that actually it's the data analysis that in general takes the longest when it comes to TEM imaging and sort of getting data from TEM. So we brought our MiniTEM system to this project, but also, and also as part of our MiniTEM system, we have a very powerful software called VS. And when this project started, when we were thinking about writing this project, we, at that point, had just released our new sort of AI pre-trained models that would come with the software. So traditionally, what we had normally was our workflow modules here to the left. These are sort of tailored image analysis boxes that you can tune and connect, and they're great for sort of non-envelope virus, so things of a fixed size. So AAVs, for example, they work really, really well, and you can really pick those up. But we knew that when it came to more pleomorphous particles, like lentiviral vectors, etc., it was more difficult for us to define what the detection modules should hold. And we found that with AI, we could actually get some really nice data on pleomorphic particles. And we knew that VLPs would be a good target to focus on. MPL brought into this their sort of renowned expertise in standardization. They also had expertise in VLP or synthetic VLP engineering. And this was together with Emiliana De Santis. She had sort of led this VAMAS project where she was looking at the size and size distribution of synthetic VLPs. So we could really use this as a sort of reference material, or as our candidate reference material for the work that we wanted to do. Yeah. And UCL holds so much expertise when it comes to development platforms, and they had a really nice project together with PT Biopharma in Indonesia and LMIC on VLP production. So this project was using VLPs as a vaccine candidate for dengue disease, and they were working on developing this platform for the production. And of course, you can see, so this was covering both the upstream processes as well as the downstream processes. And we could see that we could really come in here in this part, which is around the product characterization for quality assessment. So we got together and we got started. So the first thing we did was, of course, we put in the MiniTEM system to use, and we can already see that we're already removing several of the obstacles I've mentioned before. So we don't need to change the facility fittings around installation. The remote access and operation of the system means that both training and support is done remotely. The image analysis and the need for an expert user is also sort of lifted, as well as the speed of the analysis. But we still had these two things that we also wanted to look at, two obstacles here: sample preparation, the need for radioactivity, and the method validation. So I know that this presentation is about the feasibility study, but what we did first was our interaction voucher that we then built our feasibility study on. So I want to just mention what we did for our interaction voucher that we did together with UCL. We looked at if we could overcome the issue and the need of radioactivity and the dependence on sort of a radioactive license. So why is radioactivity used, or radioactive stain? So in negative stain transmission electron microscopy, we use stains to apply to these organic samples. And we do this to really increase the contrast and to preserve the particle integrity. And traditionally, and the most frequently used stain happens to be urinal acetate, which is radioactive. I sort of think of it as being sort of the ethidium bromide, ethidium bromide in gel, urinal acetate in nsTEM analysis. But we know that actually there are other stains that could be just as well, or actually maybe work even better, that are non-radioactive, but there wasn't really that much sort of work done on them. So we went ahead and decided to evaluate and to see if we can find something to substitute the radioactive stain. This is just the final results of it. Salome gave a presentation on this one of the Vax-Hub meetings, but we were able to find a suitable stain, a nano-W stain, which is tungsten based, and we could see that actually we got a really nice correlation, and we could actually get a very nice staining of our of the VLPs here. We were using the dengue VLPs for a reference for this, and their sort of measured size previously was around 40 nanometers, and this is the kind of size that we were getting here. So we were happy with this and the staining protocol. Now we move on to sort of the final piece of the puzzle, which is providing some kind of SOP, some sort of method validation. And this was part of the feasibility study. It was a larger undertaking. And so we set up our feasibility study design. So what we did was the work that I mentioned to you, the optimization of a non-radioactive stain here in the top left from UCL. We took that information, that knowledge, and we performed a method transfer to MPL. MPL used this information and then also worked on creating their synthetic VLPs. They also performed the data collection. They set up the framework for the analysis and also the analysis workflows. And once that imaging was done, Vironova took charge of the bit which came to the training of our AI models and our AI algorithm for the data analysis. And I will talk to you a bit more about it in the next couple of steps, but basically we go through different iterations. And when we were happy with the final iteration of our AI, we could then apply the CNN algorithm back to UCL and the PT Biopharma Dengue VLP samples to perform a protocol validation and to see how that sort of fit and how well it works in sort of a real life setting. So we were happy to see that that the method for the sample preparation with a non-radioactive stain worked really nicely on the candidate VLP reference material from MPL. To the left here under A, you can see these VLPs stained with uranoacetate. And then to the right, these are the particles. So these sort of white ones, these are the VLPs. And we can see that we're getting nice staining and we can detect them. We can detect them well. Once we had the sample preparation ready, we could then go ahead and start looking at our image capture. So TEM image capture protocol that we set down and we established. And when I say established, it means, we were looking at, well, what's the best magnification to acquire these images that gives us the right amount of information? How many images should we acquire? How many areas on the grid should we visit and should we take images for? And we did this, of course, together with MPL. And we acquired more than 300 images for each sample. And of course, we did this as well at different sites to do study tests for intra and intra-laboratory validation, looking both at sort of repetition, different sites, different users, different days. And around that, of course, with our images in place, we had to look and set out the acceptance criteria for setting up our analysis. So we had our initial data triage. I won't go through this step by step, but I want you to see that, we set up, guidelines from the vast experience of MPL and working with these VLPs of what to measure and to make sure that what we are measuring is actually VLPs and not other particles. We also set an acceptance criteria for reproducibility and repeatability. This was a sort of a standard one-way ANOVA that was used for the intra-laboratory repeatability and inter-laboratory reproducibility. And then the measurements here again, really informed from the VAMOS work that Emiliana did on the VLPs, MPL. The measurements that we set up for and that we chose was size and shape. So we looked at the minimum and maximum ferrite diameter of the particle here, and also the aspect ratio and roundness. This also to help us to verify and to steer us more towards measuring actually VLPs and not sort of other particles. And then another thing that we thought was important is the impact that some of this analysis has on time, how long it takes. How much do we gain by automating image collection? How much do we gain by automating data analysis? So with those acceptance criteria set out, we performed our image analysis and we tried different software and different detection methods. So we used the Vironova's AI software. This is sort of based on convolutional neural networks. In brief, we take a raw image, we do some annotations on these images. We basically tell it what the VLP is and we box it and we say, this is a VLP. We run several iterations of this. So we were using a minimum of 30 images with 100 particles in each of these images. And we ran a minimum of a certain number of iterations. And then we looked at how well this CNN algorithm worked on the particle detection. In the bottom left, you can see our raw image, and then followed by 2 different iterations of a VLP AI. And this was a really good sort of exercise for us as well to see how much should you train an AI, how tight do you want that detection to be. Of course, the more you train it, the more specific it is, but you still want to have that flexibility. So this was a really, really good example, and using the candidate reference material was really valuable for us. we also in MPL performed a comparison with ImageJ and their sort of standard workflow in a sort of a semi-automated manner, highlighting the automated steps and then the manual steps. So this is the kind of detection we got as an example of the detection we got using AI for our VLP candidate reference material, showing you the raw images here and the VLP segmentation to the right. I've just zoomed in here a bit, and you can see that actually we can get a pretty nice particle detection, as shown in purple. So when we were happy with that, we can then proceed and actually do the statistical assessment for the method validation. And this is, again, still working on the VLP reference material from MPL. So we could see how well we were measuring the VLP size, how well we are measuring the shape, and also looking at the data collection at different times and doing the checking the robustness of our analysis. This is just one of the examples of the sort of statistical assessment that we did. Here is our intra-laboratory repeatability assessment, where we're looking at the area, minimum ferrite diameter, aspect ratio, and roundness of our VLP particles. And this really serves as a really good basis for continued work, and we are really excited to sort of continue and furthering these assessments and see what we can do to really make the analysis as robust as possible. So, as I mentioned, we wanted to evaluate the timescale of the analysis. So here I'm just sort of plotting how long it took to work with, let's say, ImageJ, workflow analysis, manual annotation of images, and the AI software. And we can see that with the AI training, although it takes longer to develop the workflow, and actually this is sort of just letting it run overnight and these iterations, you really gain time. This is really where the time gaining comes. It comes when you are analyzing the images and the frames. So to analyze 20 images takes us 10 minutes rather than between 2 to 4 hours as it does for other image analysis or manual annotation. And this is, of course, fully automated, so you are not dependent on the specialist skills. So that was also very good to kind of lay out and get a sense of. So once we had that in place, came now the final step in the puzzle, which is our pilot tech transfer. So we took our AI model that we had trained for the synthetic VLPs, and we applied this to the dengue PT Biopharma UCL dengue VLPs. And what we could see is that we could successfully generate a really nice model for the analysis of dengue VLPs with very, very few modifications. So the training that we set out with the reference material really gave us a lot of advantage, because we only needed very little training and adjustment to be able to pick up. And I hope that you can see here the dengue VLPs that have been detected in purple. And of course, what that means is you get your quality attributes, your VLP quality attributes from your AI analysis. So you can check that you've picked up your particles. You can do your area measurements, your diameter measurements, your aspect ratio, etc. So with that sort of means that we come to our sort of summary and wrapping everything up. And this was actually done, I should say, this was done in a short period of time, just a couple of months during, hardcore COVID lockdown times. So this really provided a good basis for the continued work, and I'm really happy sort of with what we did. And I'd like to sort of summarize what I have presented and really kind of see how that links with relevance within the vaccine manufacturing field. So in conclusion, I hope I've shown you that we've been able to develop and validate a TEM analytical pipeline with defined acceptance criteria for VLPs. And we've gone really from sample preparation to data collection to statistical analysis. We've been able to evaluate the impact as well of automating imaging and analysis and looking at the benefits of scaling to larger data sets, and of course reducing the requirement for skilled operators, and thereby really sort of speeding up information on product status and increasing the accessibility of the method in LMICs and for SMEs. And I think this is something, you know, really useful to have in mind. I think we want to sort of be able to present something that you can pick up and start working with, and of course modify, but I want this to sort of connect to the final part, which is the transferability. So we did our proof of concept for this by demonstrating the applicability of the method by transferring this workflow over to the vaccine Dengue VLP vaccine product, which was developed by PT Biopharma and UCL. But we believe that this transferability could also be applicable to other VLP vaccine development initiatives and really helping you to get to those valuable insights faster. And for the future, of course, we aim to, and we really hope that we can continue this great collaboration in developing the application of the feasibility study protocol, both in the quality control environment for the VLP characterization, but also adding in different comparative studies and working further on really refining our AI and image analysis from this. So with that, I'd like to thank you all for listening. I'd like to acknowledge Emiliana DeSantis from MPL for her excellent expertise in reference materials and VLPs. Adam Dickinson and Vironova trained the AI models. Salome, of course, have been a key driver in this project and helped us as well to tie together the work that we were doing together with her project and PT Biopharma with excellent amount of information and advice, and has been a really wonderful person to work with. Ellie for her valuable information and exchange and sort of drawing from the years of expertise that she has been really, really wonderful. We also got a chance to interact a bit with Nenny from PT Biopharma, and we want to thank her for working together on the samples. And I also want to thank Rafa from the Crick Institute. So we had one of our MiniTEM systems at the Crick Institute that was going to be used for the intercomparability study, and it was great. Of course, during lockdown, this was all very complicated, and she was very helpful in making sure that we were able to perform that work. Also want to wrap up with saying that, an acknowledgement to Vax-Hub for the feasibility voucher and the interaction voucher, but also really for creating a space where you can get these excellent interactions and sort of knowledge exchange and sort of light bulb moments popping up and really facilitating collaboration and opening up discussion and allowing us to actually do something that would have taken a long time and a lot of coordination to set up in a pretty short amount of time. So with that, I will stop talking. I'm 28 minutes in, so I'm happy to take your questions, and I hope I'm still online. Yes, I am. Good.
Great. Thank you very much. That was a super presentation, and I think hopefully very clear to everyone what was happening. So I hope you can see it already happening in the chat, but I think, as we were told earlier, we'll use the chat to collect questions, and then I, as the chair, will read those out to Saba, so you don't need to worry about navigating into the chat. I'll do that for us. So I think we just go in the order of which things are coming in. So we'll start with a question from Kevin Murphy, who asks, how do you determine how many individual images are required for analysis? E.g., in the example, 300 images would the data be more accurate if more images were analysed, but at a cost of additional time to acquire this? So yeah, maybe you can comment on that.
Yeah, thank you. I think that's a really good question, and it is actually something that you kind of want to start with, right? You want to start with saying, well, how many images do I need to take? Because you don't want to take more images, and you definitely don't want to take less images. We normally take it, and actually for that, I think the candidate reference material is really good for that, right? Because you have much more control of your sample, and what you want to look at is at which point does additional data not affect your results or the information that you get. So we have done this, for example, quite often for AAVs, where we figured out that actually you need to have a specific magnification, so you're picking up a certain number of particles, and that if you go to more than 5 areas collecting 25 images in each, you're not going to see any more variation in your average and in your results that you are getting. So it sort of panels out. So that's how we kind of tend to work with this. And we also used the information from Emiliana and the work that she had done with VAMAS about, how many particles we want to be picking up and counting. So we acquired 300 images. But we settled for approximately a minimum of 1,500 individual particles. And of course, because you can also work with different concentrations. So if you have a lower concentration, then you might want to take, more images. So in some ways, also the number of particles is quite a useful and a more usable data point to kind of work with. I hope that answered the question.
No, I think it does. I think that's an interesting point, right? It says, I guess, the amount of data in the image as much as it is how many images. That's a good point. I guess, Steve, the next question from Steve Morris within the Vax-Hub, I guess takes us along a similar question. Again, maybe how to get the right amount of information in the images. So he asks, while grid preparation and staining is relatively simple, it still requires operator with some skill. How much do you find this plays a role in standardization? Is there any work on automation of grid prep and staining?
Yes. Again, a good question. And of course, you can see a method and you can say, oh, great, you just add 3 microliters. But I mean, of course, there is always more to it. There is always more to running gels and doing different. For us at Vironova, we are working on a sample preparation kit, specifically addressing that. Because we know that even applying a drop of something onto a grid can vary from person to person. So we're really trying to kind of simplify it and taking away the user variability that can be present. And of course, the more of the usability you can take away, potentially the fewer data points you might need to acquire, right? So you get a more robust data. So we are working on this currently, and we've got some sort of sample preparation kits that we are actually going to be working together with MPL. Hopefully they can be some of our beta testers for this. And yeah, it's definitely something that we are working on. Hopefully can be available.
Thanks. And maybe I'll just ask check we're not got too many questions, and we've still got plenty of time. So there's a question for myself because I think it follows on from Steve's point. I mean, obviously we hear lots and lots about AI, machine learning, solving all our problems. But looking back in some of the areas we looked at, once upon a time, we were looking at the area of mass spec and people trying to use AI and machine learning to. But some reports said, it was very sensitive to even a different piece of equipment. The model would no longer stand up, and you had to repeat the model, or maybe similar to Steve's question, if someone else is running the sample, is the data always the same, and then that confuses. Or so, are there ways around? Is your ambition that the AI model is so hands off? Any lab, you just plug it in and it goes, or does it always require some recalibration? Or what's the ambition in terms of that? Because as you said, you looked at intra lab, but then when you go into lab, I guess it gets more difficult.
Yes, and it does, and I think this is something that we are all kind of working towards and striving towards, and I think with some of these more kind of advanced techniques, there will always be more parameters that can be adjusted, right? The simpler the technique, the fewer parameters. But here we've got quite a lot of parameters, although we're looking at a 2-dimensional image, and in that sense, it's pretty straightforward when the image analysis takes place, because we're not working with fluorescence, and we're not doing any additional labeling. We're adding the samples onto a grid. We get a 2-dimensional image, and then we're basically tracing the space around it. So in some ways, there is some simplicity there that benefits us. Because there, even though you might have done, not a brilliant job at sample preparation, what we tend to do is we tend to go to different areas on the grid. So even if your sample preparation is not brilliant, there are still areas in that grid that are okay. But what takes time is for you to manually go and find those areas. So what we're trying to do is that we're trying to introduce not necessarily AI, but actually image analysis. So what we can tell the software is, anything that is darker than this, ignore it. Go to a different place. And we have taken it even a step further than that. So what we've done is that we've taken a grid square, so an area to image on the grid that we know is well stained, right? So decent staining. And then we can use that as a sort of template matching. So we tell the software to go and find areas that matches this image within a certain percentage. So you can say only ones that match 90% plus, only go and take images and do the analysis there. You can also lower that threshold, so you have sort of control of it, because some people can be extremely stringent and other not. But what it means is that you have one set of decision of what range and what conditions to do the imaging and the analysis in, and then you're saving the time by going and finding that, because not everyone produces grids at the same time. But that means that some of that variability we can move to the side and just focus on grids that really fall within, or grid squares that really fall within what we see as an acceptance criteria. So we can steer it towards those places until there is a time when grid preparation is absolutely perfect. And I mean, we have actually published this. It's a microfluidics device, and we really hope that we get a better and nice and even staining, where you're applying your sample a bit like the COVID test, right? Boop, and then it goes there, and then you get kind of your results. So while we're working on that, we're using software, not always AI, but just software, and just telling it to do different things that will help us to overcome some of those some of those sort of hindrance points as well. So yeah,
makes a lot of sense. So you can sort of speeding up that I guess the process that's quite tedious and long to do manually. Nitin, a question here now from Nitin. Thanks for the presentation. How accurate is automated imaging and analysis, I guess?
Yes. Again, so this is potentially quite subjective, right? Because you can say, how accurate is it in comparison to? So how accurate is it in comparison to manual annotation? How accurate is it in comparison to using different image analysis software, or how accurate is it comparing AI versus non-AI? So we are comparing all of those things, and we're really looking at the pros and cons and what we can improve. We can see that sometimes what you gain in time, you might lose in sort of accuracy, or what you gain in accuracy, you lose in repeatability. So it's quite a tricky one to answer, and I would say we want to continue working on this and getting more data to say to you that actually, compared to manual counting, the accuracy is like this, compared to this counting is like that. In particular, because we're working with VLPs, and I think maybe there might be a question around, well, how do you know that what you're seeing is a VLP and not something else? Comes the other thing, that even manual observation and counting, unless you've labeled it, you are guided by some of the properties of the particle to tell you it is what it is. But then again, that comes back to subjectivity and how, and then also how accurate your eye is, which is why the reference material, and I keep banging on about it, but it's really nice to have reference material, because you don't have anything else in there, right? So you're like, okay, what I see is this, and it's really well defined, and I've really checked it. There's nothing else, and my eye is telling me I can see this. So we hope to get some data about the accuracy more looking at the reference material, but I think when it comes to individual samples and so on, it needs a bit more work before I'll give an actual number. But it's pretty accurate, It's not very different from what I see with my eyes and what I pick up, and also not very different from what we do with the workflow.
Thanks. So that's great. I guess we're going in the direction that Adil was about to take us. And he's from Oxford. So he comments, seeing is believing, smiley face. So and this is probably an issue, I think many of us is maybe on the more academic side, when you're putting EM images in publications, it's a bit like looking at tea leaves in the bottom of you're trying to interpret as you were getting to. So he has some VLPs look like natural assemblies. And you mentioned proteosomes, I think, in your talk, and obviously that's really great if we can distinguish different impurities. But I guess he asks, how can this AI approach be used to confirm you are seeing VLPs, and how do you know if you're not an expert, you're not, what are you looking at? Also, can we analyze highly heterogeneous samples such as crude lysates, e.g. analyzing and comparing process samples? I guess he asks, would spiking crude lysates with reference be needed, or what sort of approach may you recommend? So, yeah.
Okay, so wow, that's a lot of questions, and that's great. And I've actually heard Adil talk. I mean, he's really a VLP expert, so I'm really happy to be honest with some of these questions. And I know that this is someone that knows a lot and has worked a lot with VLPs, and we've had discussions before about the analysis of VLPs. And so let me sort of take them one by one, right? So the first question was, how do you know that you are measuring a VLP? I guess, right? So sort of in process. And then the other one we can look at is, can you run sort of dirtier samples or samples that has lots of things in them, right? So I think we can almost have two different conversations. One of them is using a reference material is really clean, it's really pure, and can you detect those particles? And I think as I've shown you in like some of the zoomed in images, when you don't have anything else there, you're actually picking up the majority of them. And when we train our CNN, we don't just train it, we don't just tell it what the size of a particle is or what the shape of it is, but we tell it this is a VLP. So we know that some of the things that it picks up is potentially textures, things that we don't really put a specific kind of measurement to. And we know that by increasing the iteration, you can potentially get it to separate things. So we had one thing that we were doing was that when we were doing our sample preparation, there was something in the sample that created these artifacts. But we knew that these were artifacts and they had been detected before, and we didn't want to count them, but they were kind of the same size. And so what we did was when we did the training, we told it to exclude that and to kind of only focus on the VLP. It's definitely not, 100%, accurate all of the time. And that's a bit one of the downsides, I think, with working with things like exosomes and VLPs and so on. Unless you sort of label them, you are restricted to size and shape of them. The good thing is, of course, using, for example, as a bit of a bonus, is that using NanoW, that's a stain actually that works really, really well with gold nano labeling. So that would be useful as well to at least check and see, wait, I have done this labeling, now I can see that these VLPs, okay, they're labeled here, but they are within this size and they are within this shape. So that's a way for you to kind of get more confidence in your analysis and your detection. But again, I think with everything, we want to have other methods that are brought in as well, other analysis, whether it's SEC, whether it's DLS, and to really confirm something. So it's not just one method that gives you that confirmation. And then again, when it comes to the lysate question, how dirty it is, it kind of goes back to the training, especially if we're talking about the AI. So we can tell it to ignore things. And I like that, because sometimes you haven't optimized your sample and you have other things in there and you just don't want it to detect those. Of course, if if you have something that is sort of snake shaped, that's very different. It's totally going to blank that. So again, it comes to how you want to train it, how specific you want that to be. And of course, the more you train it, potentially the less flexible it will be. So there is always that to consider as well. Do you want something that is, do you want to have one AI for each step of your purification process, but that does a really good job at picking up those in that context? Or do you want something that you can run for several stages in your purification and still be able to pick that up? And then you might need to give it a bit more looseness.
Thanks. I guess related to that, are you at Vironova developing sort of different labeling techniques? Obviously, you might imagine things like lipid-based things like lentivirus, extracellular vesicles might respond in different ways to things like an adenovirus, a protein-based and VLP, protein-based VLPs. I mean, is there guidance on that? And obviously, as you mentioned in your diagram, it could be your flow sheet. It could be an area where a lot of time and effort needs to go in. How do people speed that up?
So if your question, so let me see if I understand your question correctly. So are you asking if we at Vironova are working towards guiding the interpretation and the analysis of different types of particles.
No, well, yes, I guess it's a question as well, but more on the the staining techniques themselves. You know, I imagine, you know, there's some sort of standard ones, but I guess there's potential to improve your image through the staining.
Yeah, I've got you. Yeah, yes, we do. So, you know, we're in the field of gene therapy, vaccines, drug delivery. There are some, you know, frequently used particles, and those frequently used particles. We like to try out well. So we tend to try different stains, different staining methods, different concentrations, which magnification is best to look at these particles, not to do, because sometimes what you see is really nice images, right? In a paper, a beautiful, you know, publication, but that's like one image, looks great, super magnified. But if you want to get the statistics, those large numbers, you need to sort of work in a slightly different way. And sometimes when I sit with microscopists, they say to me, well, why don't you zoom in more, you know, to see more of some of these features? But then you have to turn around and say, what is your question? Is your question to pick up those minute details, or is your question to get an overall assessment of your sample and get that statistics? Because that's sort of two different ways of doing it. You might even have staining protocols that work really well to find those lovely, beautiful things, but then you might want to use a different stain when you want to just run it, you know, daily and get thousands and thousands of data points. So the question comes back again to the application of it. And we are working with this. Most of the people at Vironova have got, you know, academic background, whether they've been microscopists or virologists. And these are things that we really like to investigate and work on. And hopefully with collaborations like this, get even more samples to try this on, because we also work with a lot of pharmaceutical companies and biotech companies, and they're also, you know, they like to keep their samples to themselves. So we can always buy material, but it's always nice to also have those real life cases, which is why, for example, the PT Biopharma work is so great, because you see that, oh, you got 8 molar urea, okay, let's work with that. You know, there are challenges and there are other things, but that is what happens in your process.
Thank you very much. So last question before I hand back. And the last question is, did you try, it comes from Renato, did you try the software with labeled samples, thinking about the analysis of final formulation of multivalent vaccines? So I guess, you know, talking about here, maybe, I don't know, glycoconjugates or other things. Yeah.
So when it comes to labeling, what we've looked at mainly has been gold labeling. And of course, then your particle looks a certain way, right? So if you want to use it to do, you know, a detection and analysis, that's fine. I mean, you can see in your microscope, you can pick it out and you can do the analysis. Now, if you want to use AI for that, the question is, will you be training it to detect particles that have got these black dots? And will that then only be used for labeled samples? Or do you want to have something that actually just looks at the samples and picks them up without the labeling? And then again, if you talk about, well, how many dots are there on this particle, and would it detect something that has 10 times more dots, or just 1 or 2 that you can observe with your eye? That's still sort of work in progress. But we know that labeling is used, and it is something that we will see, and it is also something that we want to quantify and include, but we of course don't want to have the measurements of those labels in what is defining our particle. So we don't want it to add an extra 15 nanometers and give it a, you know, a protrusion, because we need to exclude that. And we can do that with our image analysis. But we also want to see, well, what is frequently used, because as much as individual projects are really exciting in their own, it does require work and focus on those. And we're trying to do, what we're trying to do here is to really just simplify things, make things, the more applicable they are to more people, the more we try to focus on them, just to kind of broaden that scope and not go too much into one detail. But we've got an R&D group that works on sort of other projects like this, but these are longer projects. So for me, I try to look at some, you know, make things faster and just not be too complicated, really.
Thanks very much for all those insights. That's the end of our Q&A session. I'll hand back to Ludovica, who I think's got some final comments for us. So thanks from me.
Thank you.
Thank you. That was really great. Great presentation and great discussion as well. Thank you, Saba. Thank you, Dan, for chairing the Q&A, and thank you everyone for joining the conversation. That was really fantastic. Let's just, I would like just to conclude with introducing you to the next webinar and last public webinar of the platform funding webinar series, which is going to take place on the 2nd of March. And feasibility study title is Mass Spectrometry Approaches to Profile, Monitor and Localize Host Cell Protein Products in Adenovirus-Based Vaccine Manufacture. So I really hope to see you all there again. And I think this is the end of today's webinar. Thank you all for joining, and hope you're going to enjoy the rest of your day. Thank you, everyone.